We will be showing you the process of protein synthesis, which shows the central dogma of molecular biology, including both transcription and translation.
But first, let us know what is protein synthesis.
What is protein synthesis? Protein synthesis is the process by which cells make proteins. It occurs in two stages: transcription and translation.
In eukaryotic cells, transcription takes place in the nucleus. During transcription, DNA is used as a template to make a molecule of messenger RNA.
The molecule of mRNA then leaves the nucleus and goes to the ribosome in the cytoplasm, where translation occurs. During translation, the genetic code in mRNA is read and is used to make a polypeptide.
Then, these two processes are summed up by the central dogma of molecular biology, from DNA to RNA to protein.
Now, let's dig deeper and know the process of transcription.
Protein synthesis transcription.
Transcription begins when mRNA enters the nucleus.
Transcription is separated into three phases, which are initiation, elongation, and termination.
First is initiation.
Initiation starts when?
An enzyme called RNA polymerase will attach to one part of DNA where the promoter is located.
As it unzips, mRNA copies the nucleotide sequence of DNA. However, instead of thymine, mRNA uses uracil to pair with adenine. As we remembered, cytosine needs to be paired with guanine and adenine needs to be paired with thymine.
Note that only one strand of DNA is used by RNA as a template in copying the nucleotides.
Moving forward to the phase of elongation, the mRNA strand starts to separate from the DNA template, carrying with it the copied nucleotide sequence. This will let the two strands of the DNA attach again.
Lastly, in termination, the RNA polymerase reaches the part of the DNA template called the terminator. This indicates the end part of the DNA to be copied, allowing the polymerase to detach from the RNA and the DNA strands. The mRNA carrying the DNA sequence will leave the nucleus going to the cytoplasm.
As we already known what is transcription, let's move forward and know what is translation. Protein synthesis translation.
After transcription, translation happens, in which the nucleotide sequence carried by the mRNA will be used in this process.
Translation begins once mRNA attaches to a ribosome.
Just like the three phases of transcription, the same goes for the process of translation.
However, there are three binding sites in a ribosome. The P sites or peptidyl tRNA binding sites where the tRNA of a growing chain of amino acid is located.
A site or aminocryl tRNA binding site which holds the RNA that carries the next amino acid that will be added to the chain.
And E sites or the exit sites which serve as a holding place for the discharge tRNA before leaving the ribosome.
During translation, a tRNA carries an anticodon made up of an amino acid and 3 nitrogen bases, which will complement the codon of the mRNA.
Wherein a codon is a set of 3 nitrous bases in the mRNA.
Basically, this will produce the amino acid methionine, which happens in the P site.
Meanwhile, another tRNA carrying a different anticodon is waiting at the A site.
The next tRNA moves to the E site, transferring the amino acid to the next tRNA that will occupy the P site.
Translation process will continue until the ribosome reaches the stop codon in the mRNA.
The stop codons are UAA, UAG, and UGA.
The long polypeptide chain will then be released.
And after that, polypeptide chain will become proteins. And that's all for the process of protein synthesis.